speaker for today is Professor Jaume Amengual. Professor Amengual obtained his PhD from the University of Balearic Islands in Spain, where he is from. He was a postdoctoral fellow at Case Western Reserve University and at New York University. In 2016, he became a research assistant professor at the School of Medicine, and in 2018, he joined the Department of Food Science and Human Nutrition at the University of Illinois Urbana Champaign. He was promoted to associate professor in 2024. His research has been funded by the NIH and the American Heart Association. He is also the deputy editor of the ACS journal, Nutrition Science. Thank you so much, Professor Jaume, to join us here today. The stage is all yours now.
Thanks a lot for your kind presentation and for giving me this opportunity to present to all of you the work that we've been carrying out in our lab for the past years. I'm sharing now my screen, and I think you guys can see the title. Is this okay?
Yes, okay, please
perfect.
go ahead.
So today I'm going to give you an overview of the role of these compounds, which are carotenoids and vitamin A, and they are nutrients that we all eat every day, and why they are important for our cardiometabolic health. First of all, oh wait, I'm sharing the wrong PowerPoint now. Sorry. Zoom. I'm always having problems with Zoom. And when I was during COVID, I used to teach on Zoom and I always had issues. My bad. So can you see, which screen can you see now?
So it is, yeah, please, Ashish, go ahead.
We're at the Zoom screen in your browser.
Okay, sorry. I had too many things open. So now, after all these problems I'm having, I will share the correct screen. Okay. All right. So as the person that introduced me before, I'm currently the new deputy editor for this new journal, for this ACS new journal called Nutrition Science. And then in our work, we try to publish work related to nutrition and technology and things that relate to nutrients, bioactive molecules, and some also clinical trials related to bioactive compounds. As it shows here, I'm the deputy editor under Dr. Thomas Hoffman's supervision, and he's the editor-in-chief. And we do a broad spectrum of topics, as you can see in this little chart, related to gut microbiome, health, and nutrition and toxicity, and also epidemiological and other topics. Having said that, I will just jump into the topic of the day, which is carotenoids and vitamin A. So first I will give you a little bit of a background of the sources of vitamin A and why vitamin A is so important for us. Then I will tell you a little bit about this enzyme called BCO1, which is my favorite enzyme. We work on this for many, many years. And then how recently we have found a way to target the expression of this enzyme in a tissue-specific manner to see the conversion of vitamin A at different spots in your body. So vitamin A is very important, and probably you know this from school or textbooks. Vitamin A is crucial for vision. Without vitamin A in the eye, we would be blind completely. The levels of vitamin A in the eye are extremely high, actually. They are at the micromolar range. However, vitamin A has other functions in the body. One of them is helping during the embryo development, because vitamin A it acts as a hormone that allows to the formation of many, many organs in our body. It also helps in the differentiation of the immune system, and also is related to how our body regulates energy metabolism. And now I will explain you a little bit more about how this single vitamin can do all of these things. Vitamin A, in at least in the US, it comes from different sources. It can come from animal products as vitamin A itself. In the US, we do not eat too much liver or foie gras or pate, but we can get vitamin A from fortified foods and also in the milk and some fish. However, the majority of vitamin A comes from plants, and not as vitamin A as one might think. It comes as precursors of vitamin A, which in this case, these are some of the carotenoids that are vitamin A precursors. Beta-carotene, as it has two arrows here, is the main vitamin A precursor in our diet because it's the only molecule that is able to produce two vitamin A molecules. In the US and maybe in other countries, I'm not fully aware of these. This is just a slide that I use for my classes. I always want to highlight that even though in juices and these other plant-derived products, sometimes it shows vitamin A. This would be more referring to vitamin A equivalents, and that has to do because plants, as I said, and I always tell people, plants actually do not have vitamin A. They contain the precursors, which are these molecules, carotenoids. So how our body converts carotenoids to vitamin A? So we have these enzymes, 3 of them that are called non-heme iron oxygenases. And one of them, BCO1, is the enzyme that is actually responsible for the production of vitamin A. The other 2 enzymes are very important, but I'm not going to cover them today. So this enzyme, BCO1, what it does is to produce a cleavage, an oxidative cleavage into the middle of the beta-carotene molecule to form 2 retinal molecules. When that happens, retinal is an aldehyde and is very reactive and it has key functions in visions. However, what we do in our lab is to cover the effects of carotenoids and how this enzyme BCO1 and vitamin A are related to cardiovascular health and many other aspects. So when we look at the data that is out there related to clinical trials, for example, we see that there's a lot of association studies that say that these compounds, carotenoids, that we can find in our blood, in case you didn't know, but the yellow color that we have in our plasma when we donate blood is because of these compounds, carotenoids, that are very yellow. And there's many studies that study associations between the amount of carotenoids that we can find in the human plasma and cardiometabolic diseases. So in general, what we see in the literature is that higher levels of carotenoids are associated with a reduced incidence of cancer, neurodegenerative disease, mortality, and cardiometabolic disease. However, my lab, what it does is to focus on the mechanistic studies. We want to know why these compounds are very important. And out of all these diseases, I focus on cardiometabolic disease. So going back to this scheme, we have this enzyme that, as I said before, is the enzyme that is the first enzyme in our body that can produce vitamin A. Without this, we cannot do that. And retinal is specifically important for vision. It's the molecule that is more abundant in the eye. It binds rhodopsin, and then it participates in the visual cycle. A derivative of retinal is called retinoic acid. And this other molecule, it acts as a nuclear receptor activator. It actually can bind proteins in the nucleus where it will increase or decrease the expression of certain genes. And it does at different levels. But basically, the effect of retinoic acid is very different from retinal. And the functions that I mentioned before related to the immune system, energy metabolism, and the embryo development are derived from the effects of retinoic acid at the level of gene expression. And then the effects that I focus on also are derived from these effects at the level of gene expression. As you can see here, this is a picture of human tissues, adipose tissue. The middle is an atherosclerotic lesion, and then this is a fatty liver. And as you can see, the yellow color that we can see by our own eyes is because of the compounds that I've been discussing, beta-carotene and other carotenoids that we find in our body. So my research focus, it relates to the effect of this enzyme, BCO1, and how the production of vitamin A from beta-carotene can regulate these diseases. what we see is that these diseases, they have all something in common. They have, there's an accumulation at the cellular level of increased lipids, and also an increased inflammation at the level of the tissue. It happens in obesity, it happens in atherosclerosis, and it happens also in fatty liver. So this common denominator is what we want to try to figure out the effects of vitamin A. Today, I'm going to just mostly focus on the effects of atherosclerosis and fatty liver. Atherosclerosis is the main underlying cause of cardiovascular disease. The recent reports show that at least in the US, there's about 25% of people in our country that has high levels of cholesterol, and this is a very high number. And this is still the main responsible of death worldwide, not only in the US but also around the globe. Early, one of the studies that I did when I joined here at the University of Illinois was to collaborate with a partner that did some human research. And then what we did is to genotype 5 single nucleotide polymorphisms in people from which are known to participate in the activity of BCL1. So we collected DNA samples from about 500 individuals. And then what we did is to do the DNA analysis. And then we partnered it with the comparison of plasma lipid profile and food frequency questionnaires to see if there was any association between these single nucleotide polymorphisms and the activity of BCL1 and also plasma cholesterol, which is the main cause of cardiovascular health. What we observed is that this red arrow that you can see here for a nucleotide, this nucleotide specifically that is associated with an increased activity of the enzyme. We observed that the people that had higher activity of the BCL1, they had lower plasma cholesterol. So that for us was giving us an idea that increasing the production of vitamin A could have a beneficial effects in cardiovascular health. Therefore, we moved on into our experimental models, into our mice. And then for this, we use these mice that are very important for cardiovascular disease because they develop atherosclerosis as we do, as humans do. What we did is we combined 2 different mouse strains to be able to compare the effects of beta-carotene and vitamin A separately. From one side, what we did is to mix the LDLR knockout mice with our knockout mice that are BCL1 knockout mice. Therefore, these mice, as you can see here on the bottom left, they accumulate carotenoids because they are not able to cleave it and produce vitamin A. So we combined them, we created these double mutants, and then we fed them a Western diet to study the development of the disease. What we observed when we fed on one side LDLR knockout mice for 12 weeks to develop the disease, we focused mostly on plasma cholesterol and in the area of the lesion that we can observe in the aortas. When we plotted this data, the mice that were fed in beta-carotene, they are in orange, we observed that these mice had overall a lower cholesterol and also smaller atherosclerotic lesions, which was confirming a little bit what the human results suggested. However, when we did the same experiment in our double mutants, and as I say, these mice cannot produce vitamin A, we could not see any difference between the groups. Next, we kind of got interested in this other process that occurs in cardiovascular disease that is less commonly studied. Basically, when we have a disease that is developing, as you can see here on the upper right, I'll go back one more. The development of the disease happens very slowly, and then when you have an atherosclerotic lesions is when the cardiovascular events can happen. However, it is possible, it has been shown now recently in humans, and it has also been shown in very many animal models that the resolution of atherosclerosis can also occur. Basically, it's like a reverse, a healing of this disease that we develop in our arteries. So we wanted to examine the role of this BCL1 in this process. For this, what we did is to have to switch the experimental design. We used wild-type mice, and then we developed the atherosclerotic lesions first for 16 weeks by blocking the LDL receptor transiently. At that time point, we harvested a subset of mice that we call baseline here in white, where we can see the cholesterol levels being very high. And then the remaining mice, we stopped this depletion of the LDL receptor because they were weekly injections. So we stopped those injections, and then the mice became again wild type in a way. And then during that time, we fed them either a Western diet or a Western diet with beta carotene. So the mice, which is important, we never switched their diets. They never really noticed a change in the food or anything. Mice are very tricky animals. Even by changing their food, they could feel some kind of alterations and then decide to stop eating, for example. So when we did that, by stopping the depletion of the LDL receptor, the cholesterol levels normalized, they decreased. And that is one of the prerequisites for the resolution of atherosclerosis. So if you have atherosclerosis, the doctor would immediately tell you your lipids need to go down, need to be normalized. That is the first step to start healing the disease. Then we focus in this case in two main factors related to atherosclerosis resolution, which is not the size of the disease itself, is more about the stability and the amount of inflammation that these lesions have. So atherosclerosis, if it is a very big lesion, as long as it's stable in your arteries, is a good sign that it probably will not break. Therefore, we focused on these two parameters. One of them is collagen, and the other one is the percentage of macrophages. When we did this analysis, when we plotted our data, overall, you can look at here at the FDR values. We observed that there was a significant difference between all the groups when we compare them side by side, and between also the mice and atherosclerosis resolution, the orange dots. We observed that the beta carotene, when it was converted to vitamin A, was able to also help increase the stability and reduce the inflammation of these arteries. We did this same exact experiment, but with another knockout. We did here a dietary switch in another mouse model in the LDL knockouts, and then we observed similar outcomes. However, when we did the exact same experiment in BCA1 knockout mice, which are the ones that cannot produce vitamin A, as I show here on the bottom right side, and there was no production of vitamin A, we could not see any effect, any significant effect between the two groups that underwent atherosclerosis resolution. Again, suggesting that vitamin A is the key factor for the effects of carotenoids. We wanted to know how all of this process happened, so we used these reporter mice that are called FOXP3-GFP, and the reason is because FOXP3 is a transcription factor that is regulated by vitamin A, and it's very important for the atherosclerosis regression because it promotes the production of these cells that are called regulatory T cells. So to target these T cells, we wanted to eliminate them. We used an antibody that blocks and attaches to these cells and sends them for cell death. So basically what we did is to include another group here, as you can see in red, that basically served to eliminate the majority of these cells in our tissues in the mice. And then we added those antibody treatments during the atherosclerosis regression. When we did this, we observed a depletion of these Tregs in the blood and also in the spleen, which would be the last group, the one that is in orange and black. This suggested that the antibodies were effective at destroying these cells in the body. Then we looked for these cells in the atherosclerotic lesions because what they do is to stimulate an anti-inflammatory environment in the plaques. So what we did is to colocalize these two markers, C25, FOXP3, and also to do the pairings because we wanted to count the number of cells that were here, and then we could observe that there were some lesion cells that remained, and then we were a little bit suspicious about this. It seemed that the antibody was not fully able to eliminate all the cells. we actually confirmed this, but as you could see here, when we compare a mouse that has been treated with the antibody here on the right, the majority of the cells are dead. The red is gone. And then some of the cells that remain green, that is a reporter for FOXP3, remained in the tissues. Now, when we compare the number of FOXP3s in the plaques, we looked and then we counted them. We saw an increase in the regression, which was expected. And with beta-carotene, we saw even a higher number of these FOXP3s, which shows that vitamin A is a direct activator of the production of these cells. And when we were treating them with this antibody, we could see only a partial reduction of this number of cells. We also saw there was an increased activity of vitamin A in these plaques by immunostaining. This is a marker for vitamin A activity in the cells. And when we looked at the stability and at the inflammation in these tissues, we again observed there was a big difference between all the three groups that we had before. However, when we put together in the last group, the anti-CD25 anti-inflammatory group, these differences were mitigated. And we think it's because we could not completely eliminate these cells. In summary, the effects of dietary beta-carotene on atherosclerosis progression and resolution depend on the formation of vitamin A. So on one side, the progression of the disease is related to lipid metabolism, and the resolution is related to inflammation. So there are some questions that remain unanswered, unfortunately, and then this is what we've been working on this. Where is vitamin A being formed? Because this enzyme is produced in different tissues. Where it is in the intestine, it is in the liver, elsewhere. Therefore, what we did is to develop adeno-associated virus with the help of our collaborators that targets adiposity. Targets specifically the adipocyte. So what we did in this case, we used our mice, BCO1 knockout mice. We fed them beta-carotene for a very long time, for 10 weeks. And then we inject them with the virus that produces vitamin A, or a control virus that would be the GFP. Sorry, the labeling is wrong. This would be not an L, this would be an AT, which means adipose tissue. So as you can see here at the bottom on the right, we injected these viruses specifically in the inguinal adipose tissue because this virus targets adipocytes, but if you inject it systemically, it will also target other organs. Therefore, we had to perform injections in this adipose tissue, specifically in that organ. And as you can see, the organs were not yellow anymore, because there was vitamin A formation. Therefore, beta-carotene, which is orange, it disappeared from the tissue. What we observed in female mice was that these females had smaller adipose tissues compared to the control groups, which is in green. And this was associated with an increase in retinoic acid, which is the end product of one of the end products of beta-carotene. So females, smaller adiposity, higher retinoic acid levels. Interestingly, when we looked into males, these effects were gone. We could not see any differences in adiposity or the retinoic acid levels, suggesting and actually demonstrating, we did this into different cohorts, that there is sex differences in the production of vitamin A, at least in relationship with this pathway. Now we are working with a similar vector. That is, this one is for hepatocytes. Where we have created this hepatocyte-specific vector, and we have injected it in these mice, in the mice that I mentioned before, which are able to produce atherosclerosis and are also a very adequate model to study liver disease. So we have followed a similar approach. We have injected them in different routes, and then we see that retroorbital injection of this vector basically delivers the vector mostly to the liver, while intraperitoneal can reach other tissues. So therefore, we do retroorbital. This is an immunostaining showing that the virus, which is the GFP in green, is targeting hepatocytes and not targeting F480 cells, which are Kupffer cells and macrophages in the liver. We have also done the same with other cell types. We see that the virus is specific for the liver. And then, so when you look at the mice, when we inject this virus, you can see the very clear difference. So these are the same mice, they are siblings actually, and they are fed the same diet. But when you inject them with BCL1, you have this complete conversion of vitamin A, not only in the liver, but also other tissues. As you can see here, the levels of beta-carotene are decreased. These are HPLC quantifications, and then the vitamin A levels are increased. There's more vitamin A stores in the liver, there's more vitamin A signaling in the organ, and therefore there's more production of vitamin A in these organs. When we look at total cholesterol, we see also a decrease in these mice, in the ones that are injected with the BCL1. And when we looked at the atherosclerotic lesions, look how beautiful these orange atherosclerotic lesions are. They are also smaller. So mice that are injected with the BCL1, they have also smaller atherosclerotic lesions. This also occurs at another level of the aorta, at the aortic root. So we see this in different areas of the arches. And then we also see a reduction in content of macrophages that are here in red. We see another thing that we see in the blood is a decrease in the number of monocytes and pro-inflammatory monocytes when we inject the mice with this virus. And then we also see that these effects are likely mediated by reduced recruitment of monocytes to the lesions. It's basically saying that when we have vitamin A in the organs, there is less monocytes arriving to the atherosclerotic lesions, and therefore the plaques are less inflamed. Now, these are the time for my conclusions. So I hope I didn't run too fast. So what we see is that dietary beta-carotene ameliorates cardiometabolic disease outcomes in mice and in a BCL1 and sex-dependent manner. We see that beta-carotene stored in tissues. We have developed these vectors. That one of them is published, the other one is under soon will be sent for publication, and they reduce atherosclerosis. But these are effects that can be sex-dependent and not sex-dependent. And this is the lab. I want to thank everyone here that has done this work, and I will not take any questions. Thank you.
Thank you so much, Professor Amengual, for the very insightful talk.